This video includes the following chapters: Nervous System Overview, Action Potentials in Neurons, Synaptic Transmission, Neuronal Integration and Coding, Neural Circuits and Networks, and the Spinal Cord, Tracts and Pathways. Timestamps are provided in the video description for quick access to a specific chapter.
The function of the nervous system is to provide rapid communication and integration between various organs, as well as with the outside environment. It detects changes within the body and in its surroundings, and responds accordingly. Fast communication is achieved by means of electrical signals known as nerve impulses, which are generated and carried by specialized cells, called neurons. The major components of the nervous system are the brain, spinal cord, and nerves. The brain, enclosed and protected in the cranium, is the central processing center. It receives information, makes decision, and coordinates the body's response. The spinal cord, enclosed in the spinal column, functions as a communication gateway between the brain and the trunk and limbs. Nerves are cord-like structures that conduct information, similar to electricity conducting wires. They are composed of axons of neurons, the cell bodies of which are clustered in knot-like structures called ganglia. Ganglia commonly serve as relay centers, where neurons synapse and transmit information to each other. The brain and spinal cord make up the central nervous system, while nerves and ganglia constitute the peripheral nervous system. Functionally, a nerve fiber can be sensory or motor. Sensory nerve fibers carry sensory information from sensory receptors to the central nervous system, while motor nerves conduct motor instructions from the central nervous system to effector organs, the muscles and glands. Nerves that contain both sensory and motor fibers are known as mixed nerves. There are two major groups of nerves: cranial nerves and spinal nerves. The 12 pairs of cranial nerves emerge from the base of the brain and relay information between the brain and the head and neck regions. The cranial nerve 10, named vagus nerve, also communicates with internal organs. The 31 pairs of spinal nerves arise from segments of the spinal cord and innervate the trunk and limbs. Spinal nerves communicate with the brain via the spinal cord. All spinal nerves are mixed nerves. They contain both sensory and motor fibers. Typically, sensory receptors send impulses by way of sensory fibers in spinal nerves to the spinal cord, which relays the information up to the brain. The brain interprets the information and sends back instructions down the spinal cord to motor fibers in spinal nerves to reach effector organs. The peripheral nervous system can be divided into somatic and visceral subdivisions. The somatic nervous system includes sensory nerves from the skin, muscles, bones, and joints, and motor nerves that innervate skeletal muscles. This system controls voluntary muscular contractions, as well as involuntary somatic reflexes. The visceral nervous system, on the other hand, includes sensory division that detects changes in the viscera, the organs in the thoracic and abdominal cavities, and motor division that controls cardiac muscle, smooth muscle of internal organs and glands. It produces, for example, faster heart rate and breathing rate during physical exercise, and slower cardiorespiratory rate during sleep. The visceral motor division is also known as the autonomic nervous system because it is largely autonomous, acting independently of the body's consciousness and voluntary control. Neurons communicate with each other through their dendrites and axon. Generally, incoming signals are received at dendrites, while outgoing signals travel along the axon to the nerve terminal. In order to achieve rapid communication over its long axon, the neuron sends electrical signals, known as nerve impulses, or action potentials, from the cell body to the nerve terminal. An action potential is essentially a brief reversal of electric polarity of the cell membrane. Cells are polarized, meaning there is an electrical voltage across the cell membrane. In a resting neuron, the typical voltage, known as the resting membrane potential, is about -70 millivolts. The negative value means the cell is more negative on the inside. At this resting state, There are concentration gradients of sodium and potassium across the membrane. More sodium outside the cell and more potassium inside the cell. These gradients are maintained by the sodium-potassium pump, which constantly brings potassium in and pumps sodium out of the cell. A neuron is typically stimulated at the dendrites, and the signals spread through the soma. Excitatory signals at dendrites open ligand-gated sodium channels and allow sodium to flow into the cell. This neutralizes some of the negative charge inside the cell and makes the membrane voltage less negative. This is known as depolarization, as the cell membrane becomes less polarized. The influx of sodium diffuses inside the neuron and produces a current that travels toward the axon hillock. If the summation of all input signals is excitatory and is strong enough when it reaches the axon hillock, an action potential is generated and travels down the axon to the nerve terminal. The axon hillock is known as the cell's trigger zone, as this is where action potentials usually start. This is because action potentials are produced by voltage-gated ion channels that are most concentrated at the axon hillock. Voltage-gated ion channels are passageways for ions in and out of the cell, and as their names suggest, are regulated by membrane voltage. They open at some values of the membrane potential and close at others. For an action potential to be generated, the signal must be strong enough to bring the membrane voltage to a critical value called the threshold, typically about -55 millivolts. This is the minimum required to open voltage-gated ion channels. At threshold, sodium channels open quickly. Potassium channels also open, but do so more slowly. The initial effect is therefore due to sodium influx. As sodium ions rush into the cell, the inside of the cell becomes more positive, and this further depolarizes the cell membrane. The increasing voltage in turn causes even more sodium channels to open. This positive feedback continues until all sodium channels are open and corresponds to the rising phase of action potential. Note that the polarity across the cell membrane is now reversed. As the action potential nears its peak, sodium channels begin to close. By this time, the slow potassium channels are fully open. Potassium ions rush out of the cell, and the voltage quickly returns to its original resting value. This is the falling phase. Note that sodium and potassium have now switched places across the membrane. As the potassium gates are also slow to close, potassium continues to leave the cell a little longer, resulting in a negative overshoot called hyperpolarization. The resting membrane potential is then slowly restored, thanks to diffusion and the sodium-potassium pump. During and shortly after an action potential is generated, it is impossible or very difficult to stimulate that part of the membrane to fire again. This is known as the refractory period, which consists of absolute refractory and relative refractory. The absolute refractory period lasts from the start of an action potential to the point the voltage first returns to the resting membrane value. During this time, sodium channels are open and subsequently inactivated while closing and thus unable to respond to new stimulations. The relative refractory period lasts until the end of hyperpolarization. During this time, some potassium channels are still open, making it difficult for the membrane to depolarize, and a much stronger signal is required to induce a new response. During an action potential, the sodium influx, which occurred at one point on the axon, spreads along the membrane, depolarizing the adjacent patch, generating another action potential. The sodium currents diffuse in both directions, but the refractory properties of ion channels ensure that action potential propagates only in one direction. This is because only the unfired patch of the axon can respond with an action potential. The part that has just fired is unresponsive until the action potential is safely out of range. An action potential generated at the axon hillock usually travels down the axon to the nerve terminal and not back to the cell body. This is because the axon has more voltage-gated ion channels than the cell body.
Neurons communicate with each other mainly via chemical messages or neurotransmitters. When a neuron is sufficiently stimulated, an electrical impulse called an action potential is generated and travels down the axon to the nerve terminal. Here, it triggers the release of a neurotransmitter into the synaptic cleft, a space between neurons. The neurotransmitter then binds to a receptor on a neighboring neuron, generating a signal in it, thereby transmitting the information to that neuron. The neuron that releases the neurotransmitter is the presynaptic neuron, while the one that receives the signal is the postsynaptic neuron. The axon of the presynaptic neuron may form a synapse with either a dendrite, the cell body, or the axon of the postsynaptic neuron, giving rise to axodendritic, axosomatic, or axoaxonic synapse, respectively. Chemical synapses exist not only between neurons, but also between a neuron and a target cell, such as muscle or gland cell. Over 100 of neurotransmitters have been identified so far. Most of them can be grouped into classes according to their chemical structure. Major classes include amino acids, such as glycine, glutamate, aspartate, and GABA. Small peptides called neuropeptides, such as beta-endorphin and substance P. Monoamines, such as epinephrine, norepinephrine, dopamine, serotonin, and histamine. Monoamines are basically amino acids with the acid group removed. And acetylcholine, an ester of choline, in its own class by itself. Neurotransmitters are synthesized in the presynaptic neuron and stored in small sacs called synaptic vesicles at the axon terminal. Some of these vesicles are docked on plasma membrane, ready to release neurotransmitter on demand. When an action potential arrives at the nerve terminal, the resulting depolarization opens voltage-gated calcium channels, allowing calcium to flow in. Calcium causes the vesicles to fuse with plasma membrane, releasing the neurotransmitter in a process known as exocytosis. Upon binding to their receptors on the postsynaptic cell, some neurotransmitters opened ligand-gated ion channels, causing direct changes to membrane potential of the receiving cell, while others act through second messenger systems to exert their effect. Some neurotransmitters are excitatory, others are inhibitory, and for some, the effect can be either excitatory or inhibitory, depending on the receptor they bind to. An example of excitatory neurotransmitter is glutamate. Upon binding, it triggers glutamate receptors, ligand-gated ion channels, to open and allow positively charged ions into the cell, making it more positive, less polarized, and thus more likely to generate action potentials. On the other hand, GABA, a major inhibitory neurotransmitter, opens ligand-gated chloride channels to allow negatively charged chloride to enter, making the cell more negative, more polarized, and thus less likely to generate action potentials. Acetylcholine is a neurotransmitter that can be either excitatory or inhibitory, depending on the receptor present on the target cell. At neuromuscular junctions, acetylcholine, released by motor neurons, binds to nicotinic receptors on skeletal muscle cells and stimulates them to contract. On the other hand, it inhibits cardiac muscle cells via muscarinic receptor M2, causing heart rate to slow down as part of the parasympathetic rest and digest response. A neurotransmitter binds to its receptor for only a millisecond or so. It then passively diffuses from the synapse and is taken up by nearby astrocytes for recycling. If the presynaptic neuron continues to fire and release more neurotransmitters, new molecules will bind and again activate the receiving neuron. If the presynaptic signal stops coming, the transmission will eventually stop. There also exist mechanisms to actively remove neurotransmitters from the synaptic space to avoid overstimulation. Common mechanisms include degradation of the neurotransmitter by an enzyme present in the synapse, and reuptake, where the neurotransmitter is transported back by a transporter protein to the presynaptic neuron for reuse. A typical neuron receives not one, but many synaptic inputs at a time. Moreover, these inputs may have similar or opposing effects on the receiving neuron. Neural integration refers to the process in which a neuron integrates all input information and decides whether or not to produce action potentials. Neurons are polarized, meaning there are different electrical charges inside and outside the cell. A typical neuron has a resting membrane potential of about negative 70 millivolts. The negative value means the cell is more negative on the inside. In order to generate an action potential, membrane potential must reach the threshold of about negative 55 millivolts, meaning the inside of the cell must become less negative. The cell becomes less polarized or depolarized. Any voltage change in that direction is called excitatory postsynaptic potential, EPSP, because it makes the neurons more likely to fire. EPSPs are brought about by excitatory neurotransmitters. On the other hand, inhibitory neurotransmitters change the membrane voltage in the opposite direction, producing inhibitory postsynaptic potential, IPSP, making the cell hyperpolarized and less likely to generate action potentials. A neuron can receive both excitatory and inhibitory inputs simultaneously. It's the balance between EPSPs and IPSPs that decides whether or not a neuron will fire. A typical EPSP produced by a single action potential in the presynaptic neuron is around 1 millivolt and lasts about 15 to 20 milliseconds. To reach the threshold, the postsynaptic neuron must receive multiple EPSPs nearly at once. There are two ways this can happen. Temporal summation, when a single presynaptic neuron fires repeatedly at short intervals, producing EPSPs so closely to each other that each EPSP occurs before the previous one decays, allowing their effects to add up and reach the threshold. And spatial summation, when multiple presynaptic neurons fire nearly synchronously to produce multiple EPSPs in the postsynaptic neuron. All action potentials produced by a particular neuron are identical, and yet they underlie the ability of our brain to perceive all the vast and complex information in and around our body. Just like Morse code, which uses simple dots and dashes to encode an unlimited number of messages, our brain uses action potentials to encode complex sensory information, a process known as neural coding. Firstly, the brain differentiates between different stimuli by activating different sets of neurons. For example, different odors activate different sets of olfactory neurons in the olfactory nerve. Sounds with different frequencies stimulate different nerve fibers in the auditory nerve. Different taste sensations excite different taste cells. Secondly, information about the intensity of a stimulus can be conveyed by activating receptors with different sensitivities, stimulating a smaller or larger number of neurons, or producing a lower or higher firing frequency in one neuron. Thirdly, aside from the firing rate, other characteristics of the action potential pattern may also encode information. Various coding methods have been proposed, including temporal coding, which emphasizes the precise timing for each action potential spike, in particular, the first spikes produced by a population of neurons. Phase coding, in which neurons transmit information by firing at different phases with respect to a background cycle. And burst coding, where neurons communicate in a burst of spikes with characteristic spike numbers and intervals between them. Neurons work together in groups called networks, each concerned with a particular body function. These networks can be very large and extremely complex, but they are typically built from several simpler motifs called circuits. In a feedforward excitation circuit, one neuron activates a second neuron, which may go on to excite a third, and so on. This is a way to relay information and activate a pathway. On the other hand, a feed-forward inhibition circuit is formed when a neuron stimulates an inhibitory neuron, which then inhibits the next neuron. This circuit is used to shut down a pathway. Feedback excitation is when a neuron is activated by any of its downstream neurons. Once the first neuron in the circuit is stimulated, the circuit is self-perpetuating until this first neuron is switched off. This is a way to maintain a persistent, long-standing response to a brief stimulus. Conversely, feedback inhibition is when a neuron is inhibited by any of its downstream neurons. The purpose is to terminate or limit excitation in a pathway after an effect is achieved. This setup works like a thermostat, shutting down cooling or heating once the desired temperature is reached. Feedback inhibition is very common. It is used also in biochemical pathways to maintain homeostasis. Convergence is when one postsynaptic neuron receives signals from multiple presynaptic neurons so it can integrate different inputs to make a decision. Conversely, divergence is when one presynaptic neuron communicates with multiple postsynaptic neurons, coordinating their action in a network. Lateral inhibition is when a neuron excites inhibitory neurons, which then inhibit the neighboring neurons in the network. This circuit is used, for example, in the retina to provide edge enhancement. It is also responsible for some visual illusions. Feedforward excitation and inhibition circuits often work together to control opposing muscle groups to produce movements. An example is the knee jerk reflex. When the knee is tapped by a hammer, the resulting stretching of the muscle causes sensory nerve endings in the muscle to fire. The axons of these sensory neurons activate motor neurons in the spinal cord, which then stimulate the extensor muscle to contract. This is an example of a feed-forward excitatory circuit. However, in order for the leg to extend, not only the extensor muscle must contract, but the flexor muscle in the back of the leg must also relax. This is accomplished by a feed-forward inhibitory circuit. As it enters the spinal cord, the axon of a sensory neuron gives out two branches. One branch excites motor neuron of the extensor muscle, while the other activates an inhibitory interneuron. This interneuron then inhibits the motor neuron that controls the flexor muscle, keeping it relaxed. Similar circuitry setup underlies voluntary movements controlled by the brain. Basically, the brain sends out signals to two feed-forward excitatory circuits that control extensor and flexor muscles simultaneously. But because these two circuits inhibit each other via inhibitory interneurons, their activation occurs alternately and not simultaneously, producing contraction in one group of muscles and relaxation in the other. When combined with the spinal cord crossed extensor reflex, the movement of the other limb can also be coordinated in a similar manner, so that a person can alternate between the two legs while walking. The branching of the sensory neuron's axon in the knee-jerk reflex is also an example of divergence. In fact, one sensory neuron can have multiple branches activating multiple motor neurons, causing multiple muscle fibers to contract at once. Interestingly, convergence also occurs in this network. When the knee is tapped, many sensory neurons are stimulated simultaneously, and they all converge on individual motor neurons to amplify the response.
The spinal cord is the communication gateway between the brain and spinal nerves, which innervate the trunk and limbs. The cord is a long, thin tube of nervous tissue, enclosed in 3 membranes of the meninges, which in turn are protected within the bones of the vertebral column. The 31 pairs of spinal nerves arise from the cord and emerge from the vertebrae. The spinal cord extends from the brainstem to the level of upper lumbar vertebrae. In the lower lumbar and sacral regions, nerve roots descend within the spinal canal before exiting, forming the cauda equina. In cross-section, 2 types of nervous tissue can be seen in the cord: a butterfly-shaped central core of gray matter and a surrounding white matter. The gray matter contains cell bodies and dendrites of neurons. This is where neurons synapse and transmit information to each other. The white matter, on the other hand, is made of bundles of axons and serves to conduct information up and down the cord. These bundles are organized into specific groups with specific functions, forming the so-called spinal tracts. Spinal tracts are essentially high-speed cables, each carrying a certain type of information in a one-way traffic, between the spinal cord and a certain area in the brain. All tracts occur on both sides, left and right, of the cord. Ascending tracts conduct sensory information up to the brain, while descending tracts convey motor instructions down the cord. Some tracts cross over to the other side of the cord before they reach the brain. They convey sensory information from one side of the body to the other side of the brain. When this happens, the information is said to be transmitted contralaterally. Tracts that stay on the same side all the way are said to conduct information ipsilaterally. Spinal nerves are mixed nerves. They contain both sensory and motor fibers. These fibers are separated shortly before they reach the spinal cord. Sensory fibers enter the cord via the dorsal root, while motor fibers exit via the ventral root. A sensory pathway typically involves 3 neurons. First-order neurons detect stimuli and transmit signals to the spinal cord. The axons of these neurons form sensory fibers that enter the cord via the dorsal root of spinal nerve. Inside the cord, first-order neurons synapse with second-order neurons, which ascend a specific tract to the brainstem, or further up to the thalamus. In some pathways, first-order neurons ascend the tract to the brainstem, where they synapse with second-order neurons, which continue to the thalamus. Third-order neurons conduct the information the rest of the way to the sensory cortex. A motor pathway usually involves 2 neurons. An upper motor neuron starts in the motor cortex or brainstem, and a lower motor neuron continues from the brainstem or spinal cord. They conduct motor instructions down along a specific descending tract. The axons of lower motor neurons exit the cord via the ventral root of spinal nerve, where they continue as motor fibers to effector organs. The spinal cord is also responsible for fast, involuntary responses of skeletal muscles, called somatic reflexes. Reflexes are essentially automatic and do not require input from the brain, although the brain is informed and aware, usually after the fact. A somatic reflex involves a reflex arc composed of a somatic receptor, a sensory neuron, an interneuron, a motor neuron, and an effector muscle. Some reflexes are, however, more complex and require multiple pathways, as well as central coordination from the brain. For example, when someone steps on something sharp and lifts their injured leg, the other leg also must react to keep balance or the person would fall over. This involves multiple muscles and requires contralateral pathways at several levels of the cord, as well as movement coordination from the brain.
Are you a fan of Alila Medical videos? If so, you will love our new Alila Academy. Based entirely on our highly effective animated mini lectures, our courses are designed to follow a typical syllabus in each subject. Each topic is extensively illustrated and comes with quizzes to test your understanding, downloadable PDFs for a quick review, as well as images for you to use in school projects or presentations. We are known for producing highly effective animated videos that explain the most difficult concepts in record time. We have no doubt that our courses will be a lifesaver for both students and teachers alike, so do come check them out. Alila Academy. Clear, concise, effective, with extensive visual resource. No one explains like Alila does.